Hello, my name is Joshua Mugabi. I welcome you to Biology Lights. And in this session, we are going to look at cell chemistry. Apart from food, what other substances do cells need to take in? Cells need to take in water and salts in addition to food. Water has a high heat capacity, what we are referring to as the thermal capacity. This is an advantage in living cells because A. Any rise in temperature is small in comparison with the amount of heat absorbed by a cell. B. Any rise in temperature is large in comparison with the amount of heat absorbed by a cell. C. The 75% water in a cell does not retain a lot of heat. D. Any change in temperature will cause a cell to heat up or cool down quickly. And the LIPID, it is A. Any thermal capacity or a high thermal capacity means that any temperature rise is small in comparison with the amount of heat absorbed. This helps to protect the cell against extremes of temperature. Give 3 examples of cell structures which contain structural proteins. There is the cytoplasm, the cell membrane, membrane systems in the cell, the nucleus, and mitochondria all contain structural proteins. What is the other type of protein in a cell? Enzymes are the other type of cell proteins. Name the chemical elements present in a protein. Proteins contain the elements carbon, hydrogen, oxygen, nitrogen, and sulfur. What name is given to the subunits which make up all proteins? All proteins are composed of subunits called amino acids. A protein molecule which is denatured has A. Split into smaller molecules. B. Changed its shape. C. Combined with another molecule. D. Been diluted. It is B. A protein which is denatured has changed its shape. What kind of substance is a lipid? A lipid is a fat or oil. It may be combined with other substances, for example, phospholipid or lipoproteins. In a cell, where are lipids found? Lipids are found in cell membranes and other membrane systems in the cell. Some cells may have food reserves in the form of lipid droplets. What are the two types of chemical compound which combine to form a lipid? Lipids are formed from the combination of fatty acids with glycerol. What elements are present in a lipid? Lipids contain the elements carbon, hydrogen, and oxygen. Name 4 examples of compounds which are classed as carbohydrate. Sugar, we have glucose, fructose, maltose, sucrose, starch, glycogen, and cellulose are examples of carbohydrates. What elements are present in carbohydrates? Carbohydrates contain elements carbon, hydrogen, and oxygen. Write the formula for glucose. The formula for glucose is C6H12 or 6. If that symbol represents a glucose molecule, draw A, a maltose molecule, B, part of a starch molecule. A maltose molecule will be like that, and B, part of the starch molecule, will appear like that. Select the most appropriate words for the list below to complete the following paragraph. All cells contain dash, which are dash, and act as dash, which dash chemical reactions. The reactions do not dash the dash which can take part in further reactions. The words to select from, we have substances, proteins, enzymes, catalysts, speed up, use up, slow down. our statement will be all cells contain enzymes, which are proteins and act as catalysts, which speeds up chemical reactions. The reactions does not use up the enzyme, which can take part in further reactions. Enzymes will usually react with only one substance. This can be explained by the lock and key theory. If this theory is correct, which of the following substances represented by P, Q, R, and S would be acted on by enzyme A? Using the lock and key model, enzyme A is most likely to react with the substance Q. If an enzyme-controlled reaction normally takes place at 10 degrees Celsius, in general terms, how will the reaction be affected by A, a fall in temperature to 2 degrees Celsius, B, a rise in temperature to 20 degrees Celsius, C, a rise in temperature to 65 degrees Celsius. If an enzyme normally works at 10 degrees Celsius, then a fall in temperature to 2 degrees Celsius will slow down the reaction. Then part B, a rise in temperature to 20 degrees Celsius will speed up the reaction by 2 times. And part C, a rise in temperature to 65 degrees Celsius will denature the enzyme and stop it working, though the reaction may speed up at first. If an enzyme is denatured, why does it no longer work? An enzyme which has been denatured has changed its shape and will no longer combine with its substrate, the substance it acts on. The graph shows the rate of an enzyme reaction at different level of acidity or alkalinity, that is pH. From the graph, what is the optimum pH for this enzyme? A, pH 2, B, pH 7, C, pH 10, D, none of these. It is B. The optimum pH is 7 because the rate of reaction is greatest at this pH. A protein digesting enzyme, when mixed with the starch solution, would A, have no action, B, produce amino acids, C. Produce glucose. D. Digest starch. It is A. A protein digesting enzyme would have no effect on starch. Select the most appropriate words for the list below to complete the following paragraph. All enzymes are produced inside dash. Enzymes which do their work outside cells are called dash. Enzymes which do their work inside cells are called dash. Most of our digestive enzymes are examples of dash enzymes. The words to select from: animals, extracellular, intracellular, cells, digestive, nuclei, catalysts. So our statement will read, all enzymes are produced inside cells. Enzymes which do their work outside cells are called extracellular. Enzymes which do their work inside cells are called intracellular. Most of our digestive enzymes are examples of extracellular enzymes. Give 2 examples of chemical reactions which are catalyzed by enzymes in the course of brewing. In the course of brewing, enzymes in the grain catalyze the conversion of starch to maltose, and enzymes in yeast catalyze the conversion of maltose to alcohol. What does the enzyme catalase do? Catalase speeds up the breakdown of hydrogen peroxide to water and oxygen. Substance A is being investigated to see if it is an enzyme. When substance A is mixed with the substance B, a reaction takes place. A control experiment is conducted using a sample of A which has been boiled. Part A. Why is boiling used as a control? And part B. If the reaction still worked after A had been boiled, what might be your interpretation? Part A. Boiling denatures enzymes. If a substance still works after boiling, it cannot be an enzyme. Part B. If the reaction still worked after A had been boiled, either A is not an enzyme, or if it is not necessary for the reaction. In an investigation, to compare the rates of which starch is being broken down by an enzyme. A. What test is used? And B. How do you know when the reaction is completed? A. The test for starch is iodine solution, which goes blue. And part B, when no blue color appears after adding iodine, all the starch has gone and the reaction is complete. Hello, my friends. Thank you very much for participating in Biology Lights. Please do subscribe.